Hi, everyone. My name is Evie Tiplady, and today I'm going to talk to you about the characterization of the impact of fire on soil fungi communities. Under the direction of Dr. Charles Hauser, I had the opportunity to explore the question of whether or not soil fungal communities are affected by forest fires, and if so, how. So I'm really excited to talk to you about the data that we collected and the research we did. However, I feel like there is a huge importance to discuss soil fungi, their role in the soil and the ecosystem they exist in, as well as the natural fire cycle. So let's start there. So the composition of soil is not just one microorganism or another, but rather a community of microorganisms that form what's called a microbiome. Their composition, this community includes archaea, bacteria, and fungi, along with a number of other microorganisms. Specifically looking at fungi, we want to see what their responsibilities are, or what their role in this ecosystem is. And what we know is that they're extremely important for nutrient uptake, signal sending and receiving, and pathogen chemical defense. In the case of nutrient uptake, the interactions between the plant and the fungi are kind of like a bartering system in that each has what the other wants. In the case of fungi, it has nitrogen and phosphate, what the plant wants, and the plant has carbon, which the fungi wants. These two kind of exchange, allowing for each to coexist and thrive in the ecosystem it's in. Additionally, soil fungi are extremely important for signal sending and receiving. We can think of fungi as this broadband internet connecting a number of plants to one another. They allow for the communication of everything from who's depleting in nutrients to where disease-ridden plants may be located. A third and final responsibility of soil fungi is pathogen defense. They have the ability to release chemical if there's any threat to the fungi or the plant in the form of bacteria, competing fungi, or any other pathogens present. So now that we have an understanding of the role of fungi in soil, I want to talk a little bit about the natural fire cycle and its importance. So the natural fire cycle is important in that it produces biological diversity, new habitat, and it maintains the overall health of the ecosystem. So what happens is old growth develops over time, and that old growth is big and it's mature. And it means that less sunlight is reaching or accessing the forest floor. And as a result, the vegetation on the forest floor has what's called a high fuel load, meaning it's more combustible and more likely to ignite. And when it ignites, these forest fires move through. And post-fire, we have the opportunity for new growth to develop. When new growth develops, it's because there's much more sunlight available to the forest floor, and there's different soil nutrients present. What we're interested in studying is what the fungal communities present in the soil look like post-fire. So I want to talk a little bit about the forest fire that we got our results did our research our research after. And that's the Bastrop Complex forest fire. This forest fire occurred from September through October of 2001. It destroyed 32,000 acres, 1,600 homes, and it killed 2 people. It was extremely destructive also on the Lost Pine Forest, which is located in and around the Bastrop State Park. This forest contains specifically lobo pine, which are predominantly found in southern locations. This is actually an image of the forest fire. So now I want to move on to some more statistically significant images that kind of represent locations, the severity of burn and the expanse of burn. So on the left, we can see this image representing locations of high, moderate, and low burn locations. And on the right, we can see an image representing the bounds of the fire itself, and then highlighted in red, the Bastrop fire within the Lost Pine Forest, affecting those lobo pine. So with more of a statistical understanding, and now that we have maybe an understanding of the natural cycle of fires and soil fungi, we can get into the research. So we knew we wanted to study fungi, soil fungi post-fires, but we had to have a way to identify different types of locations with the changing severity of fire. So our samples were taken from permanent locations within Bastrop State Park that have been used for past soil sample collection for data that happen to be located within regions of high, low, and no burn. Those classifications were created by the Forest Service in that they use soil composition, temperature, and vegetation present in order to classify locations as high, low, or no burn. So now we can talk about the process that went or occurred in order to get our results. And the first step was DNA extraction and isolation. So we have these soil samples that contain DNA, but we need to isolate the DNA. In order to do that, a chemical wash needed to be performed that allowed us to pull out the sequences of DNA. After which time, we had to have a way to access those sequences. And that meant we had to send our data off to Mr. DNA, a company that uses Illumina technology in order to sequence our DNA. They then sent us our results, and we used QIIME2 to analyze the DNA. So this is kind of my step in the whole process and how I worked with Dr. Hauser to get our results. We used QIIME2 in order to visualize through alpha and beta diversity. Alpha diversity being within a sample and beta diversity being between samples. So at this point, you're probably kind of wondering how we were able to get just fungal DNA. We talked about the fact that the microbiome of soil doesn't contain just fungi, but it has rather all these other microorganisms. But so we had the opportunity to isolate the DNA of fungi through using what's called these internally transcribed spaces. These are sequence portions within ribosomal DNA here and here that act like a barcode. They allow us to identify the sequence belonging specifically to fungi. And they're used for both identification of fungi and then classification of the fungi. In the case of this research, we use ITS region 1. So now that we kind of have a concept of the overall process that we went through in order to get our results, we can talk a little bit about QIIME2. So QIIME2 is a platform or a pipeline that we use within a cyber infrastructure called Cyverse. So Cyverse, being the cyber infrastructure that we use, is a National Science Foundation's funded infrastructure that's led by the University of Arizona with the Texas Advanced Computing Center and the Cold Springs Harbor Laboratory to analyze, collect, and share data. It's the platform that allows us to run complex bioinformatics processes like QIIME2. So QIIME2 pipeline has a number of steps that include demultiplexing, clustering, denoising, analyzing, taxonomy, and diversity metrics. So the first step, demultiplexing, involves separating the sequences via the barcode data. In other words, using those internally transcribed region spaces that we've identified to separate the fungi from everything else. Following which, we're going to cluster those sequences based on similarities in ITS regions with one another. After we've clustered the DNA, we want to do what's called denoising, and this is kind of a quality control method. It allows us to remove any sequence errors in the data. So once we've done that, we can assign taxonomy. And in order to assign taxonomy, what we're doing is comparing our regions, our sequences in our samples to a known fungal database. In this case, the UNITE database. So this database, in comparison to our samples, the sequences, allows us to assign taxonomy. After which, we're able to measure the differences and similarities within and between samples. This image on the right is kind of to give you a sense of what a step in this process looks like. This is the demultiplexing sequence step. And I make the joke that running commands in a computer is kind of like asking a small child to do something. It needs to know the who, what, when, and the where, and the why. And the same goes for a computer. And that it needs to know exactly where the information is coming from, what you want it to do with it, and where you want it to go. I don't think I really understood that process before I started doing this research, but now I think I have a good grasp on it. And with the continued patience of Dr. Hauser and myself, we were able to generate some results. So the first result I'm going to talk about is a Bray-Curtis biodiversity plot. This is a principal analysis, coordinate analysis. And this is a very holistic diversity measure in that it's showing variability, but it's not showing what's causing the variability. And what I mean by that is often when you think of axis 1 and axis 2, we think that each represents an independent variable. But rather in this case, we want to think of each axis as representing just an amount of variability. And the big thing to draw attention to here is axis 1. If we look up from axis 1, we can see the clustering of 3 separate groups occurring in low, high, and no burn sample types. We don't know from this graph, we don't know what's causing this variability and this difference amongst the samples, but rather we know that something between the differences or variation between the data here is separating these or clustering these locations into separate groups, which is really interesting. So to kind of get into why or how this clustering is variation is occurring, we can talk about enrichment data. So in this graph, we see the enrichment data is representative of locations of high burn and no burn. So we have the genus on the x-axis and the abundance of the phylum on the y-axis. So positive values are representative of an enrichment of phylum in locations of high burn, versus negative values are representative of enrichment in no burn locations. Bassamicata, a phylum, is present in green. If we look here, we can see it's predominantly enriched in locations of no burn than in high burn. Another phylum that's extremely important here is phylum Assamicata. If we look over to the left of center, we can see the presence of some specific genus within the phylum Assamicata that are present in location of high burn, but are rather lacking or non-existent in locations of no burn. These are some factors that may be contributing to that variation between locations of high, low, and no burn. Next, I want to talk about some family level alpha diversity, and specifically looking at the fact that the percentage of sequences coming from each family is very different within each sample type. We see that the families present, represented by the colors here, are very different from the families represented in low or high burn. Specifically, I want to draw your attention to Aspergillacea in low burn. It's a representative of a much higher percentage of the sequences present here than in the other location. So these differences also feed in the samples, feed into the overall, that original picture, having some groupings of differences between no, low, and high burn locations. To get a little bit more specific, we can now talk about some classes of fungi that are predominant in certain locations. So argumocytes are a class of fungi responsible for decomposing dead organic matter and contain sautrophic fungi, which mineralize and stabilize soil moisture. Additionally, they produce external spores. So these fungi or class of fungi was predominantly found in locations of no burn. By comparison, this class of fungi, plasmocytes, grow in extremely barren environments. They contain mycorrhizal fungi, which connect plants and soil, and their spores are internal. They, oppositely, establish soil dominance post-fire in locations of high burn. So, again, these are some features that may be causing some separations or differences between that original graph in the samples as types and locations of high, low, and no burn. So with that, I kind of like to wrap it up in that we can conclude or understand that the soil fungal composition within locations of high, low, and no burn post-fire is different. They contain some different, not only diversity, they contain different fungi, and the quantities of those fungi is different. You're probably wondering really what the practical implications of that are. And we discussed the soil fungi's jobs or responsibilities earlier in this presentation, and they're responsible for a number of important interactions plant to plant or plant to soil. And if we don't take the time to understand how they work and how they work and their responsibilities, we're not going to be able to do our reforestation efforts as good. We think often when we consider reforestation about the trees. In this case, in this image, we have a replanting of global pine in the lost forest. but we don't always think about the soil fungi composition, but we should, because from this presentation we saw that the soil fungi are responsible for a many number of jobs and are directly influential in the plants that they interact with. Some further questions that we kind of need to understand going forward are, what are the duration of these post-fire effects? So how long are the soil fungi composition different than its original before or pre the the fire composition? Additionally, how we may go about, or if we should go about, reestablishing these original communities of soil fungi. So with that, I'd like to acknowledge Greg Kiersey, the Regional Natural Resource and Wildland Fire Coordinator in Bastrop, and the Texas Parks and Wildlife Department, for not only facilitating the collection of these samples, but also the funding that was created in order to sequence the samples, the DNA that we isolated. Claire Barnett was hugely important in collecting samples and isolating the DNA, and Dr. Hauser has been fantastic mentor for me and this entire project. I cannot thank him enough for all the work he's done for me and this whole project. I also would like to thank all of you for coming to my presentation. I look forward to hearing all your questions.